Welcome back to UC Davis Live, COVID-19. Last month, we saw the first shots of COVID-19 vaccines being given to healthcare workers at UC Davis and elsewhere. What's in these vaccines? How do they work? How effective are they? And is this really the beginning of the end of the pandemic? Well, joining us today are two UC Davis experts on vaccines and vaccination. Stephen McSorley is professor in the Department of Anatomy, Physiology, and Cell Biology at UC Davis School of Veterinary Medicine. He's also director of the Center for Immunology and Infectious Diseases at UC Davis. His research focuses on the immune response to pathogens, especially memory T cells, that allow the immune system to remember previous infections. Dr. Stuart Cohen is professor of internal medicine and chief of the Division of Infectious Diseases at UC Davis Health. He's been leading UC Davis's participation in clinical trials of COVID-19 vaccines. Thanks to you both for joining us today.
Thank you.
Sure.
So before we get started, I'd just like to remind viewers that this is a free webcast. You can watch it live and watch it after the live broadcast on the UC Davis Facebook, Twitter, and YouTube channels. We're aware of others posting links saying that you have to pay to watch this video, so please do not click on those links. This is a free webcast. So Stephen, let's start with some basics. In general terms, how do vaccines work?
Yeah, so what a vaccine's trying to do is trying to fool your immune system into thinking you've previously had this infection before. So if you've had an infection, you normally respond better the second time around. And the vaccine's just trying to trick your immune system into thinking that's already happened. So it does that. There's like three key components of how what we call immune memory. And they are, you have an incredibly low frequency of cells in your body that would recognize any pathogen. And so a vaccine's trying to expand that very small population. So the second time around, there's many more cells to fight against an infection. The second thing it's trying to do is that these low frequency cells have very low function. They can't really combat the pathogen effectively. So the other thing the vaccine's trying to do is to provide these cells with the information they need to combat the infection. And the third thing they're trying to do is the location. These cells in our body are swimming around our lymph nodes and our blood, but they're not really in the sites where we would normally encounter a pathogen. In this case would be the lung. And so after vaccination, what happens is you have a high frequency of these immune cells that recognize infection. They have immediate function so they can combat the pathogen, and they're actually in the right location. They're sitting in the lung, or if it's a different vaccine in another body tissue. But so those three things have to change to respond better to a second infection, and a vaccine's trying to do all those things.
That's really interesting. I didn't realize that about the location. Is that something that vaccines have always done, or is that something that's new with these new vaccines?
No, that's the way it's always been. So if you, I mean, it's kind of interesting, like in your body, you have, like I said, the capacity to respond to any infection. And I tell the students, like, if you think about it, that means if an alien race comes from outer space and they bring with them a pathogen, you actually have the capability to respond to that pathogen no matter what it is, as long as their life form conforms to our normal science. So, and that's because your body, these cells, the T cells and B cells in our body, their receptors are randomly generated. So most of your immune system is completely useless to you. It's directed against things that don't exist or things that you'll never encounter in your life. So the trade-off for the immune system to have that amazing function to respond to anything is that these cells have to be at very low frequency. Otherwise, we would just be a big bag of a lymph node walking around on legs. So we have these low frequency cells, and they're further constrained as they only move around between the lymph and the blood. And then they wait until they're activated because there's so few of them, most of them will never be involved in our lives. But once they're activated and they're told you're necessary for survival, then they expand, they gain function, and they move into the tissues.
That's really interesting. I want to remind people who are watching, if you are watching us live and would like to ask a question, just leave it in the comments section and we'll try to work in as many as we can. So Stephen, some of these vaccines are based on mRNA. How is that different from a traditional vaccine?
Yeah, it's different and in some ways, and it's not so different in other ways. So there are many different types of vaccine. We can spend a lot of time talking about that. But one typical vaccine that's commonly been used in the last few decades is a subunit vaccine. What all that means is you take a component of the organism, be it a bacteria or a virus, and you inject a person with just that component. And usually those are proteins that we artificially make from the pathogen. So this is the way hepatitis B vaccine works. And so this is a protein injection into your body to induce this response from these lymphocytes that swim around our lymph nodes. Now, all the mRNA vaccine is doing is, instead of injecting people with protein, you're injecting people with the information that the cell needs to make that protein. So it's just one step back up in the kind of way proteins are made. So mRNA is a natural constituent of all our cells in the body. We have the information for life contained in the nucleus and the DNA. And then it's kind of like a library, like the nucleus contains all the books that you'd ever need to encode life. But at any given time, some of those library books will be withdrawn because these are the ones that are needed at that particular time. And they're found in the cytosol of the cell, and they're made into proteins. So all the vaccine's doing is trying to inject that mRNA into the cytosol of our cells, deliver a library book that we don't have in our nucleus artificially, and then make the protein that we need. So in some ways, it's a different strategy. In other ways, it's kind of similar to the subunit vaccines that we're used to.
But one of the differences is that it's not like an egg-based or anything like that, or, like people had allergies to some other vaccines in the past. This is totally different in that.
Exactly. So the egg-based vaccines are because that's a live vaccine that's grown in eggs. And so that's not part of this. This is a purely synthetic generation of mRNA in a culture system in a laboratory.
How quickly do these vaccines become effective, and how long do the effects last? Do we have any data on that at this point?
Yeah, I mean, the last question is probably the most important one. And as everybody knows, we've only had this virus around for a year, so we really don't have a good answer to that question. There was just a paper came out last week from a group down in San Diego who reported memory responses to natural COVID infection persist for up to 8 months. But we don't know because, this is so recent, we don't really know what the longevity of this protective memory would be. Stu can probably give more information, but my understanding of the clinical trial for the Pfizer vaccine is within about 5 to 10 days, you start to see evidence of protective immunity after the first shot.
So as far as how long the effects last, I mean, it's just kind of like an ongoing observation. We just have to wait and see.
Yeah, exactly. I mean, there's no reason to believe that it would be really transient, because that's not the way usually memory works. But until we're actually at the point where we know that information, I think it's kind of premature to know how many times we'd have to get this vaccine, or whether it would be. Some infectious diseases give you long-term protection for the rest of your life, and others wane over time. And in this particular vaccine, it induces a strong antibody response that's thought to be the main protective aspect. And it's not known whether that will wane over time and we'll have to get a booster, or whether there's other components to our immune system that would take over, like T cells, and would then provide additional immunity.
Right. Okay. Quick reminder again, if you're watching this live and would like to ask a question, just leave them in the comment section and we'll work in as many as we can. Stu, were you going to jump in there? I'm sorry.
I was just going to mention that I agree. So by 10 days, you start seeing the placebo patients and the patients getting vaccines starting to separate, where the placebo patients continue to get infected and the patients that get vaccine don't get disease. I don't think we know whether these vaccines actually prevent infection. They prevent disease. And then the second point is that these clinical trials are really designed to go out 2 years. Now, whether people actually get followed for 2 years, because everything's changing, right? The placebo arms, everyone's getting vaccinated. So how long it lasts is part of the idea of doing one of these 2-year trials. Potentially, if something untoward happens late, that would be identified, but mostly it's to make sure that people are still immune 2 years. The other thing that has come up, right, is that the virus is changing, and whether these mutants end up getting to the point where the vaccines wouldn't be as effective anymore, is unclear. At this point, the mutations that have been identified seem to be at least neutralized in the laboratory by the antibodies that are generated by the vaccine. But ultimately, what we don't know is if this is going to end up being like influenza vaccine, where we have to get vaccinated at a given time period, probably not annually, but maybe every 2 years or every 3 years, or whether this will be like measles, where you get a second booster at some point and you never have to get it again. That's unclear, but because these are so-called RNA viruses, part of the way they survive is to mutate. And so they're going to keep changing. The more people that get infected, at least in my way of thinking, the more people that get infected, the more diversity we're going to see in the virus. And if it gets to the point where the diversity allows it to escape the immune response that the vaccines generate, then we're going to need to modify the vaccines.
And again, the vaccine just helps people not get sick from the vaccine. So if you're saying, the more people get it, the more variants there will be.
Yeah.
It's almost like a whack-a-mole thing that we're just constantly going to have to keep on top of.
Well, it may well be, right? I mean, you're used to that with influenza already. The thing is that I think many of us actually believe that the vaccine prevents infection. We just don't have data for that yet. Mm-hmm. We know people don't get sick, and the so-called endpoints in the clinical trials were patients diagnosed with clinical COVID-19. So they weren't routinely testing everybody, or at least they haven't analyzed the data if they were. And so right now what we know is that people don't get sick, and the number of people that get severe disease after getting vaccine is negligible. And most of those occur within the first 10 days after you get the first dose. And then beyond that, it becomes very unlikely that somebody actually gets symptomatic disease. But part of the reason that we're telling people to continue to mask and continue to maintain social distancing and minimize exposures is because we don't really know whether transmission can still occur. And that is something that needs to be identified. But we also know that it's going to take a while for these vaccines to roll out. And in the meantime, there are a bunch of people who have been getting infected. And when this so-called B.1.1.17 strain showed up in Great Britain, it was thought, well, sort of the same way people thought when they first saw cases in Wuhan, China, that it wasn't everywhere already. These mutations, I'm sure, are everywhere already. It's just a matter of looking for them. And so, right, and again, this isn't a surprise. This is how HIV works. This is how Hep C works. This is how flu works. RNA viruses, they develop genetic diversity by making mutational changes every time they multiply. And some of them do it faster than others. This is sort of an in-between one. It doesn't go as fast as HIV or Hep C, but it does move. And normally there has to be some motivation for it to keep some of these mutations. And those motivations would be surviving people's immune systems.
Right. Now, UC Davis has been involved in trials of the Pfizer, Moderna, and now Novavax vaccines. How are those three vaccines different?
So I think Dr. McSorley did a great job of describing what RNA vaccines are. And the Moderna and Pfizer vaccines are both RNA vaccines. The Novavax is a more traditional subunit vaccine. The way these are made actually is a piece of the viral protein called the spike, which is what every immune response targets, is put into an insect virus called baculovirus that then infects moth cells. And the moth cells produce the spike protein. that is then attached to some type of matrix. It's some secret scientific formula matrix that no one knows exactly what it is, that presents the spike protein. And then there's another molecule in the vaccine called an adjuvant, which is a lipid, which is what coats the RNA vaccines and helps generate the immune response. And so it's a more traditional style vaccine. It's a similar type strategy as was already mentioned. Every single vaccine, no matter what platform they use, are all targeting exactly the same thing, with the expectation that the outcomes of all the vaccines are going to be excellent, since we already know how good the responses have been to Pfizer and Moderna's vaccines.
Do you have data on the efficacy of the Novavax vaccine? Is it as high as the Pfizer and Moderna?
So the only thing that's been reported so far are the phase 1/2 studies have been reported in the New England Journal, I think 2 weeks ago or 3 weeks ago, that demonstrate strong antibody responses and actually demonstrate some T-cell response as well, that I suspect Dr. McSorley can explain much better than me. And so the expectation is that it engenders protective antibodies, and therefore it's likely to work. But the phase 3 trial is what we're embarking on right now. And I think there have been a little over 9,000 people enrolled nationally so far out of the 30,000 that are being targeted. So hopefully the study will enroll quickly and the data will be available, because the more manufacturers, the more good vaccines we have, the better the distribution is going to be. And when people ask whether I would take Pfizer or Moderna preferentially, my answer is I take the one that they have.
Okay, I think, go ahead, please. Stephen, were you going to chime in there or no? No.
No.
Okay, so you kind of answered, Stu, you kind of answered one of our viewers wrote in a question. Ed S, who's watching on Facebook, says, I'm a 72-year-old male with underlying medical conditions. Which of the two vaccines, Pfizer or Moderna, do you advise I receive as soon as they're available to my cohort? So you're basically saying whichever one you can get.
Whichever one comes first.
Okay. And he has a follow-up. He says, given the answers to the following 3 questions, do you advise I receive 2 full doses with recommended time between doses as soon as available, without waiting for 6 to 12 month additional longitudinal data on vaccine efficacy?
Yes. So I think, there's been a lot of discussion about whether you really need 2 doses or not, and whether the timing is totally critical or not. And the answer is, I don't think we can mention those questions. But when we look at the antibody responses, the second dose does really boost the antibody responses quite significantly. And so the expectation is that a booster dose is probably indicated, other than the Johnson & Johnson vaccine, which is the only one that's a single dose vaccine. And is 21 days or 28 days absolutely fixed? I don't know the answer to that, but I know that the best way to take it is the way that it's been studied, because that's where it's been demonstrated to be effective. And I think freelancing is probably not a good idea right now. And ultimately, I think there's been this discussion that maybe if we just get first doses into everybody, we can get more people vaccinated. But we already don't know how long the immune response lasts. I'm not sure that mixing it up with single doses is going to make any sense, and ultimately is going to do anybody any good.
Yeah, I can see both sides of that. It's a difficult one, because if COVID-19 is raging throughout your community, getting into as many arms as possible with 1 dose would seem to be, I can understand why some governments have chosen that strategy. But as Stu said, the only hard data we have on protection is from the clinical trials. that have a particular dosing regimen and a boosting strategy. So it would seem to be a little risky to diverge from that. Generally, a second dose will always increase the response you get from the first dose. And that goes back to what we talked about originally, is the longevity of these vaccines. It may well be that a single dose would give sufficient protective immunity, but that will probably it wane from a lower set point than you would have got if you had two shots. So I think in some, like I know from the UK, because that's where I hail from originally, they've gone to a strategy of just prioritizing a single shot. And I think they're banking on the fact that generally it doesn't really matter when you give a second shot. And as we know, other vaccines don't have such a short time window between the primary and boost. But as Stu said, I think we would be best advised also to follow the clinical trial as closely as we possibly can, because that's where we actually have data.
Right.
Yeah, I think probably you would agree that you don't want to get them early.
Yeah.
Right. If you're going to miss the timing, you'd rather do it on the long side rather than on the short
exactly.
side.
Is there any sense whether the Pfizer or Moderna vaccine, one or the other, can protect against these variants? Is one better than the other for variants?
So to the best of my knowledge, the only laboratory data is with the Pfizer vaccine, and it demonstrates that the major change in those variants that changes the attachment and increases the transmissibility, those mutations are effectively neutralized by the antibodies that are generated by the Pfizer vaccine. There's no real reason to expect that it's going to be any different, because all of these are so-called full-length spike proteins. So it's not just picking a little piece of the spike protein, which is maybe more prone to being affected by one of these mutations. What do you think, Steve?
Yeah, I mean, from the limited data that is available, these, it's just a laboratory test looking at antibodies and how they affect the virus grown in a culture dish, which may not be truly the same as a live infection, but it does look in that kind of limited situation that the vaccine protects against these variants. The other thing that's missing from that analysis, though, is all of these laboratory assays look at antibody responses, and as Stu said, that looks to be the main means of protection that we're trying to induce, but also there are T-cell responses, and those T-cell responses don't depend on these conformational determinants of blocking the virus binding to cells. So it may be that even if in those limited laboratory assays you saw an example where the antibody from a vaccinated patient did not block the virus, there may still be a protective effect of the vaccine inducing T-cell responses, but we won't know until, I think as Stu indicated, what mass vaccination is going to do is put tremendous selective pressure on the virus to try and evade this response. And it's basically an experiment of nature to see whether that can happen and how good the virus is at doing that. Thankfully, it doesn't mutate as much as other viruses, but it has the potential to evade these vaccines, and we'll find out.
Another viewer, Duffman857, who's watching on YouTube, is asking, should you get the vaccine if you have a cold or aren't feeling well?
I'll let Stu take that.
Yeah, thank you. I don't think there's really any evidence that you shouldn't. I think depending on how ill you are, because some people do get significant effects from the vaccine, including fevers. And so if you feel really crummy at the time, I might want to wait, just because you don't want to feel crummier if you happen to be one of the people that get a reaction to the vaccine. But I don't think intrinsically there's any reason not to give you the vaccine if you have a cold.
Okay. Kelsey, watching on YouTube, is asking, well, we've kind of answered it a little bit, but how long do the antibodies from the vaccine last? Are the antibodies the long-term protection, or is it the memory B and T cells that are the protection?
Yeah, we don't know because, like I said, we don't have the history of this, and normally we would have a longer time frame to do these kind of studies. The suspicion would be all immune responses fade over time as we age. Some of us who are old enough to have got the smallpox vaccine when we were kids, we probably still have like antibody responses to smallpox and limited T cell responses. Whether they would be protective at this stage is not clear, but they will fade over time, and we don't know. What we know is that antibody responses are protective, and in animal studies, it looks like T cells also play a role. One of the reasons we think that is because if you look at animal model studies, it looks like if you transfer antibodies prior to infection, they're very, very effective at preventing disease. And that would be the situation the vaccine's trying to emulate, generate strong antibody responses, so you get lower disease. But in studies of patients where you try to transfer antibodies, it looks to be less effective, and that would be transferred after they've got the disease. And so that is a little bit of a red flag that would say that maybe antibodies are not the only thing that would be protecting you against infection, and also T cells would be required. Or maybe I'm just biased because I'm a T cell immunologist, but I think T cells operate in a completely different way, and we can't measure them well. One of the reasons I mentioned earlier is that your memory T cells are in the tissues. So how can you sample T cells from someone's lung to know how effective those responses are? It's very, very difficult. You can take blood and look at what's in the serum, and that's antibody responses, and you get some memory T cells and B cells there, but it's very difficult to fully assess effective T cell responses that would be in the tissues.
So I have one comment, and then I have a question for you too, Steve. So the comment is that there actually is a clinical trial that we participated in that gave the monoclonal antibody to people who were exposed to someone with COVID to see whether it actually prevents infection. So it was a placebo-controlled trial. It was the combination monoclonal antibodies that are made by Regeneron. And the idea was that if a household member was infected, we gave the antibodies to the uninfected household member, and then that person is followed for whether they get infected or not. And this was a multi-center national trial that enrolled a lot of people, and we may have data, and that would again support what you're suggesting, that antibodies are likely to prevent disease, but maybe don't work so well once disease is established. The one question I would have is that we often focus with T cells and memory B cells on what's called an anamnestic response, which means that we may not be able to detect antibodies immediately, but as soon as the virus is there, everything springs back into action and you get a big response. And we bank on that for things like hepatitis B. But one of the differences between hepatitis B and the SARS-CoV-2, the virus that causes COVID-19, is the incubation period. So with hepatitis B, the incubation period is a month and a half on the short end. And so for something that has a short incubation period like this, is it likely that T cell or B cell memory would be able to spring into action quickly enough to be able to control the infection?
Yeah, that's a good question, Stu. So memory is a complex topic, and especially T cell memory. There are many subtypes of T cell memory in the body, some of which are in the blood, some of which are in the tissues. But I recently discovered a population of these, what we call tissue resident memory cells, and they sit in tissues and they don't recirculate. And so it's only been recently appreciated that these cells exist. And it seems like these cells are so effective at responding that they actually respond before the innate immune response is called into play. So innate immune responses are the part of our immune response that actually usually protect us from a lot of different things. They're very kind of low tech. Many cells in the body have this capacity to respond to things that we encounter every day, but they're not important in memory. But in the memory situation where you have these tissue resident memory cells, they respond even quicker than this very primitive innate immune responses. So yeah, in that situation, Stu, I would imagine that if you have tissue resident memory cells in the lung, they could respond within hours to not just to kill virally infected cells, but they actually also recruit other cells into the tissue, just like an innate immune response would do. And so it's almost like immunity backwards. Usually you have the innate immune response first, then the adaptive response. But when in a situation where you have these tissue resident cells, it's flipped and you have the adaptive response calling in the innate immune response. So, I mean, I'm sure people will work this out in the next couple of years about how exactly this works, but there is a role for T cells in that very fast secondary response, but we just don't know how important that is in these patients.
So we have a couple more questions from viewers. We've kind of touched on this one, but I think it bears repeating because I think it's a little confusing for people. So Debbie on Facebook is asking, can a vaccinated person spread the virus without knowing that they have the virus?
At this point, I will say yes.
Yeah, I would assume yes at this point.
Because again, the virus is just helping to mitigate any kind of illness the person actually has. You still have the virus, you could still be spreading the virus.
Yes,
Yeah,
correct.
I mean, sorry, go ahead, Stu.
No, I was just going to say, until we know that how effective, so we know it's 95% effective at preventing disease. What we don't know is it's 95% effective at preventing infection. And if it is, then the chances of transmitting after you get vaccine is pretty low. If it turns out that it's 50% effective in preventing infection, but 95% effective in preventing disease, then you have a 50-50 chance that you might still be able to spread. That's the bit of information that isn't available at this moment to be able to answer that. So I would assume the answer is yes, until proven otherwise.
Yeah, and it might be frustrating that we don't have the answer to that question, but I think these clinical trials were designed to just try and keep patients out of the hospital, to try and get this pandemic under control. They weren't designed to look at viral transmission, and that information will be, I'm sure, included in further studies. But until we know that information, I think everyone should assume that they may be still able to transmit the disease even after vaccination.
David from YouTube is asking, should you get the vaccine if you have recovered from a COVID-19 infection?
So the recommendations are yes. And the way I've looked at this is twofold, and I look at it as a clinician. So Dr. McSorley may look at it differently as more of a scientist. But as a clinician, I know that the immunity lasts at least 3 months, and as mentioned, this more recent paper says 8 months. So I think people can wait at least 3 months, and that's what we're recommending. That allows other people to get vaccinated while you're already immune. And also, just anecdotally, we've seen several people who have gotten vaccinated who had COVID, who had a more severe reaction to the vaccine. So I think spreading it out allows for more people to get immune and more people to get vaccinated. And then that would boost your immunity after a natural infection and potentially prolong the immunity in the person who gets vaccinated. So 90 days, and then I think it's okay.
Okay. Gina on Facebook is asking, knowing that a virus likes to mutate, as vaccination rates increase and herd immunity begins to kick in, what's the likelihood of this accelerating the virus's potential to change and adapt to the vaccination?
I would say high. I mean, whether that, I mean, the virus is already mutating. What the vaccination is going to do is select out mutations that could potentially evade the protective response of the vaccine. How big a problem that is, we just have to wait and see. There's really what we know is, as Stu has already said, is this virus It doesn't mutate as much as other RNA viruses because it has a proofreading mechanism that it corrects errors when it's inside a cell, and other viruses don't have that capability. But we won't know until we get there, unfortunately. What we know just now is this vaccine protects us against the virus that's out there. Whether it's going to protect us against the virus of the future, there's no way to really know that.
You know, the one other thing I would mention is this. So part of the concept of herd immunity is that instead of 3 people being infected for every person that's infected, it becomes 10 people being infected for another person becoming infected. And so if the virus stops circulating and is cut off, that's the way to cut back on replication. And the analogy I use, which is not population based, but with our HIV patients. So when we were giving them antiviral drugs that didn't totally suppress virus from multiplying, they developed a whole bunch of mutations. But when you give something that stops it cold, then no mutations develop. So in this case, if we slowly roll out vaccine, that's like the perfect way in which to generate these mutants. And if you get people vaccinated quickly and get this pandemic under control, then you have a fighting chance. I suspect there are going to be escape mutants, but I think that the more bodies it's in, the more likely they are to show up. Every time it multiplies, it gives it a chance for a mutation to show up. So you want it to just stop multiplying.
So when do you think we will see the vaccine making a difference in the pandemic? Is it at this point basically a logistics issue, getting it out to as many people as possible as quickly as possible?
I think so. I mean, the predictions based on these models is that you need probably at minimum 60% and probably closer to 75% of the population immune to be able to really stop this pandemic totally. So we're a ways away from that.
I think there is anecdotal evidence, from like, I think even UC Davis Health, now that there's a large vaccine coverage, Stu can correct me, but the number of staff off sick has decreased markedly. So you're starting to see an effect of the vaccine even now, but that's in a very small population. It just, as the vaccine spreads out, you'll see that move through the whole population of California and the US.
Right. Well, we have herd immunity within our walls right now. And I think that's part of the point. But once we leave our environment and get into the Maine of Sacramento, there's not herd immunity there.
Sarah on Facebook is asking, is there any reason for people to be worried about long-term effects 5 or 10 years down the road from any of these vaccines?
Oh, from the vaccines, there are long-term effects from COVID. Of even some people who get mild disease have this strange long COVID disease that we don't quite understand. That could be prolonged fatigue and various catalog of symptoms. And I think that's going to be something for the future. And it would be another reason for, even if you're fit and healthy and you feel pretty good, you should get vaccinated just in case you become one of these individuals. Yeah, but I don't know, there's no long-term consequences of the vaccine that have shown up in the clinical trials, right, Stu?
Yeah, that's correct. But again, it's been not really long term.
Yeah,
I think the evidence that long-term effects occur from other vaccines is negligible. And so I don't expect that this would be the case. And I would re-emphasize what Dr. McSorley's saying, which is basically the disease is bad. The vaccine is not bad, right? You may get transient side effects, but you're not going to be sick 10 months from now or 12 months from now like you are more likely to be if you actually get COVID-19, right? The risk benefit is so highly in favor of the benefit of the vaccine that I would not really be concerned about that.
Yeah, I mean, I understand people being cautious about anything these days, but I think vaccines have to be seen in the light of our history. You know, like if you think of smallpox, 300 million people died of smallpox in the 20th century, and it was eradicated in the 1970s because of vaccination. We used to, if you talk to your grandparents, they'll tell you about people with polio. They'll tell you about measles was rampant throughout our community in the 1950s and 60s, and vaccines have taken that out. So vaccines are a way to, it's not, the cost benefit analysis is not, it's not like a, it's so far in favor of vaccination that it's, I understand people's concern, but everyone should get vaccinated.
By the way, I think you're calling me old, because I had classmates with polio when I was in grade school, and I had every one of those childhood diseases. But there were mortalities from measles, and there were mortalities from polio, and all those things. And the herd immunity that we have from those vaccines is what allows people who don't believe in vaccines to get away with it.
Exactly.
We talked about long haulers a minute ago. We got a question from Craig on Facebook who's asking, should people suffering from long-term effects from COVID-19, so-called long haulers, should they get vaccinated?
I don't think I can answer that question right now. I would say that it's unlikely to provide a ton of benefit. I think I'd have to look at that as a case-by-case basis. I'd have to evaluate.
We know so little about what's causing these long COVID. It's really difficult to know.
Right. How has UC Davis been contributing to COVID vaccine research since the pandemic started?
Go ahead.
Well, I was just going to say, so we here, I'm at the Center for Immunology and Infectious Diseases, and we work on, vaccines and immune responses against a variety of infectious diseases. When the pandemic hit back in February, we started a collaboration with the School of Medicine to try and grow the virus first year, because we had facilities to grow dangerous viruses. And we also have the Primate Center next door. And so it really is it's been a great collaboration between experts in immunology on this side of the causeway and clinicians over in the School of Medicine, Primate Center researchers next door, the vet school, which is where I belong, my faculty home is. And so I think we are actually ideally poised to do some research on COVID. And so we grew the virus initially for diagnostic assistance. And then we have also researchers who have been developing a monkey model of infection to trial various treatments, including antibody therapies. And then we also have other faculty here who are looking at the long-term effects of COVID on the immune response. And those basic science studies are going to be important down the line. So that's how the basic sciences have got engaged. And I think actually has been a fruitful way to draw the basic sciences into the clinical realm. And I think our faculty have enjoyed that experience so far, despite the fact it's been a stressful situation and long work hours for many.
Yeah, so I would echo what you're saying, in addition to making a couple other comments. So we actually have a couple members from the vet school and from the Primate Center who attend our clinical conferences every week now, including our journal club and our case discussions, basically because we're all locked on Zoom right now. And so it's easy for them to do it from Davis when we are doing our conferences here. So we have gotten a lot of feedback, and we get immediate responses from basic scientists to some of our questions when they relate to the mechanistic issues that us clinicians are not quite as familiar with. The other things that we have contributed is we were a high enroller in the Pfizer vaccine trial. We are in the midst of enrolling the Novavax trial. We've been actively involved in all the treatment trials. We were one of the first places to use remdesivir. We've been involved in all the randomized trials that have got remdesivir FDA approved. And so there are a lot of areas where we have been involved. We've also done work looking at transmission within the healthcare facility. And then we were responsible for identifying the first case that was transmitted in the community. After some convincing, it took a couple days, but they finally ran the samples on somebody who hadn't traveled anywhere.
Right.
And I think the other aspect is that we're poised in the primate center because we have monkey colonies and we have aged individuals. So we have the potential to do studies that can be done in many places where we can look at the effect of COVID on this issue of age and why age is such an incredible risk factor for disease and underlying more comorbidities and how they relate to developing severe disease. And so I think that is one of the advantages that Davis has in terms of ongoing research for the future.
We're going to have to wrap up in a couple of minutes for time, but I do want to get to a couple more questions. We've got a couple of questions about herd immunity. Both AJ on Facebook and Adam on Facebook. So they're asking, can we achieve herd immunity with a vaccine that doesn't prevent transmission or infection? If you can still be infected and transmit, you aren't really immune, right? Can you kind of like tease that out a little bit?
Yeah, I mean, I wouldn't say that would be herd immunity. I mean, herd immunity, if you have, and the estimates vary because we don't, and the R number for the virus has changed recently based on these variants, so it's difficult to say, but maybe 75 to 85% coverage might be needed for herd immunity. But the principle of that is to protect the vulnerable people in our society. There are people that can't get vaccinated, people who are in cancer therapies or other immune therapies where or maybe they have underlying immune deficiencies where they couldn't respond to infection, but they're protected by everybody else getting vaccinated. But if those are vaccinated people were sharing the virus, that's no protection at all. So I don't think that's really herd immunity, right, Stu?
Yeah, I agree. I look at it as sort of, again, I think we have to see what the data show, but one of the assumptions would be that the reason that people don't get systemic disease when they are infected after they've received vaccine, even if they get infected, is because there's less virus present, and it may still make it less transmissible. I think there are too many open areas to address that. We are all expecting that the vaccine is actually preventing infection and that we are going to reach herd immunity in that way. But I don't think that we can answer that immediately at this point.
Yeah, I mean, let's just put it like, it would be unusual for a vaccine to prevent disease without having some effect on transmission. But we're just trying to be cautious because we don't have the data yet.
Right, right. Okay, and just to wrap up, I mean, is the knowledge that we've gained from COVID-19 vaccines, is that going to help us, for the next pandemic, which hopefully will not happen for a really long time?
Yeah, I think so. So I just want to point out that even though these are the first two mRNA vaccines that have been utilized, the predecessor to COVID that was called SARS, which was caused by a virus called SARS-CoV, and this is SARS-CoV-2, those vaccines were being developed at that time. Identifying what the target of the immune system was identified, and that was the first shot at trying to develop RNA vaccines. And luckily, SARS sort of faded on its own, but I think that technology is what allowed this to happen so fast. That's why people feel that this is so condensed and that it can't possibly be safe. The tools to put these vaccines together have been developed over the course of the last 15 years. And so, of course, I would think that this will prepare us better for the next go round.
Yeah, and I mean, there's experiences and technology and from this pandemic that will be useful next time around. The thing I would like to emphasize is like, we need people. Like I have a training grant that trains young people to work on infectious disease animal models from the National Institutes of Health. And I think we need more of that. We can't predict where the next pandemic is going to come from, but whenever it appears, we have to have clinicians and scientists who have broad experience in dealing with the type of issues that will arise in the pandemic. And so this issue of pandemic preparedness hopefully will get a lot more attention from our government in the future, and will prioritize the training of scientists and clinicians in this kind of area, which has historically not been a popular area.
It's popular because it's interesting, but it's not popular because of other reasons. And so I think that's important. I also would emphasize that this also teaches you that public health departments and other bits of infrastructure to contain infections can't be stripped to the bone and expect an immediate response to getting things under control. And so things that seem to be money not well spent by some people is coming back to bite us right now.
An ounce of prevention, right? I mean, being prepared ahead of time is all that matters, right? Well, listen, I want to thank both of you so much for coming on today. And I want to let you know, we've gotten so many messages of thanks from people watching for your sharing this accurate and up to the minute information. Thank you so much for coming on today.
No problem. Happy to do it.
Thank you.
Dr. Stephen McSorley is professor in the Department of Anatomy, Physiology, and Cell Biology at UC Davis School of Veterinary Medicine. He's also director of the Center for Immunology and Infectious Diseases at UC Davis. And Dr. Stuart Cohen is professor of internal medicine and chief of the Division of Infectious Diseases at UC Davis Health. Until next time, I'm Satirius Johnson, and this is UC Davis Live COVID-19.